Hello, my name is Matthias Mon, and I work in the Fuller Lab with Dr. Obadon. Today, I want to share a little bit about our work on the effects of genetic adjuvants on immune responses to DNA vaccines in a non-human primate model of HIV infection. First, some background on human immunodeficiency virus. This virus is the causative agent of AIDS. One of the reasons that it's difficult for our immune system to fight this virus is because it attacks some of the very cells that would help to combat it, those being the CD4 positive T cells. Despite significant advancements in antiretroviral therapy over recent years, HIV remains a significant threat to global health and is still an ongoing epidemic with more than 690,000 deaths every year and 1.7 million new infections every year. The current treatments remain expensive and inaccessible in some of the areas in the world which are most affected. An effective vaccine has not been identified, but advances in vaccine technology, especially in nucleic acid vaccines, may be the key to finally doing this. DNA vaccines, like all nucleic acid vaccines, supply our body's cells with the genetic information that they need to express viral antigens and to mount an immune response without actually having to be infected by the virus. Some advantages of DNA vaccines include reduced cost, as our own cells are manufacturing the viral proteins, the capacity for rapid reformulation. In case the virus mutates, you can just change the vaccine sequence. The pathogen is not included, so there's no risk for infection. The viral proteins are expressed from host cells, just like they would be in a real viral infection, so the antigen structures are very accurate. In addition to the normal B-cell mediated antibody responses that we normally think about when it comes to vaccines, DNA vaccines also induce T-cell responses, which can play just as important of a role. Finally, their increased stability makes their storage and shipping more practical. This in contrast to mRNA vaccines that must be kept at extremely low temperatures. So this is a diagram showing a little bit of the mechanism behind DNA vaccination. DNA plasmids encoding viral antigens are injected into the cell using a gene gun. This technology loads DNA onto gold atoms to create projectiles that can be fired through multiple membranes into the nucleus of cells. From there, the viral protein is expressed and can be sensed by immune cells, which mount a specific immune response to the antigen. For our multi-antigen encoding, or MAG DNA vaccine, these antigens include HIV GAG-PUL fusion protein, as well as a full-length side chain antigen. FLSC is also a fusion protein consisting of the HIV viral entry protein, GP130, bound to the CD4 receptor. This fusion exposes conserved viral epitopes that are normally buried within the glycoprotein structure. One issue with DNA vaccines is that they tend to have low immunogenicity due to minimal activation of host immune sensing pathways. Thus, we see minimal inflammation, the innate immune response is not sufficiently activated, and the cytokine secretion and antigen presentation necessary to facilitate an adaptive immune response does not occur. The solution to this problem is to include genetic adjuvants in the vaccine formulation. These are plasmids that encode host proteins that are often intermediates in the host immune signaling pathways. When these are expressed, they stimulate and direct the immune response with the goal of granting the host immunity from future infection with the pathogen. The adjuvants that we used in this study include IL-12, referred to as the gold standard genetic adjuvant due to its demonstrated ability to significantly increase the immunogenicity of DNA vaccines in both animal and human clinical trials. This protein also plays an important role in the activation of T cells, among other things. IL-18, which is a pro-inflammatory cytokine and promotes the recruitment of innate immune cells, and IRF7, a transcriptional activator of type 1 interferons, which are critical for activation of the host antiviral response. With this information in mind, we form the hypothesis that combining genetic adjuvants will provide improvement in multiple antigen-expressing DNA vaccine immunogenicity, leading to better protection from HIV infection. First, we identified a combination of adjuvants that increased the immunogenicity of a DNA vaccine encoding HIV antigens in mice. We looked for high IgG2A to IgG1 antibody ratios, indicative of a bias toward Th1 T cell responses rather than Th2, as this is indicative that the adjuvants are inducing the type of immunity that will be protective against viral infections like HIV. The adjuvant cocktail that had the highest and most consistent responses included IL-12, IL-18, and IRF7, the effects of which were discussed previously. Mice models are the first step in characterizing the effects of HIV treatments in vivo, but these models are limited, in part because mice do not replicate the effects of HIV on non-hematopoietic tissues. An improved model looks at SIV infection in Asian macaques. SIV is a closely related virus to HIV, and its disease progression mirrors HIV closely, making it one of the best models for predicting if potential HIV and vaccine candidates will be effective in humans. We took the promising adjuvant cocktail identified in mice and moved on to a non-human primate study. We had 2 randomly assigned study groups of 8 rhesus macaques, a control group that received only the standard IL-12 adjuvant, and a trial group that received the adjuvant cocktail. Multiple antigen-expressing DNA vaccinations were given at week 0, week 4, and week 12, with a protein vaccination boost at week 12. Blood samples were taken every 2 weeks for the first 20 weeks. From week 20 to week 27, the animals were challenged with a low dose of SIV rectally, and blood samples were taken every week to monitor infection status. Our prediction was that the adjuvant cocktail group will have increased anti-SIV IgG antibody titers and be more resistant to viral challenge than the IL-12 alone control group. So, to measure the relative immunogenicity elicited in the 2 groups, we quantified antigen-specific antibody titers at a baseline time point 4 weeks before the first vaccination, a time point 6 weeks after the first vaccination when the animals had received 2 doses of the DNA vaccine, and a time point 16 weeks after the first vaccination when the animals had received 3 doses of the DNA vaccine and a protein vaccination boost. We quantified the antibody titers from these 3 time points using enzyme-linked immunosorbent assay, or ELISA. To do this, we first coat the well of the assay plate with the viral antigen, then overlay plasma from the non-human primates. Antibodies in the plasma that are specific to the virus will bind to the antigen. Next, we wash the plates, then overlay reagent antibodies specific to rhesus macaque antibody FC region. These are conjugated to a peroxidase enzyme. We then wash the plates again and introduce a substrate that becomes pigmented when oxidized by peroxidase. Lastly, we measure the absorbance at 450 nanometers and compare with a standard curve to determine the antibody titer. Here are the results from that assay. As you can see, the SIV GP130 specific antibody titers in the adjuvant cocktail group were significantly elevated relative to the control group, both at week 6 and at week 16. This is indicative of increased immunogenicity and supports our hypothesis that combining adjuvants has this effect. P-values were calculated using Mann-Whitney non-parametric tests. It is also noteworthy that the magnitude and variance in the antibody titers were increased after the 3rd DNA vaccination and protein boost. Despite this increased immunogenicity, however, the adjuvant cocktail group was less resistant to viral challenge, as shown here. 4 out of the 8 animals in the adjuvant cocktail group were infected after just 1 challenge, compared to just 1 in the control group. All of the animals in the adjuvant cocktail group were infected after 5 challenges, whereas the control group were infected one by one, actually leaving 1 animal uninfected after 9 challenges. So to summarize, despite increased IgG antibody responses, the group that received the 3 genetic adjuvant cocktail was more susceptible to infection than the control group that received the IL-12 only. So from this, we can conclude that our hypothesis that this adjuvant cocktail would increase immunogenicity of the vaccine was correct. However, rather than being protected, the animals that received the cocktail were more susceptible to SIV infection. An observation that could guide our analysis moving forward is that interferon gamma ELISpot analysis of peripheral blood mononuclear cells from the study animals showed that T cell responses in the adjuvant cocktail group were elevated relative to the control group. Other studies have shown a link between high levels of induced CD4 positive T cell recruitment and a decrease in protection due to increased viral targets at the site of viral challenge. If you remember from my first slide, HIV specifically targets T cells, so the idea behind this is that if too many T cells are recruited to mucosal tissues as part of the vaccine-induced immune response, this can increase the targets for HIV and increase the chances of getting infected. To see if our data is consistent with this hypothesis, we would like to analyze mucosal samples that were taken as part of the study using flow cytometry, which has the capability to divide cells out by which receptors they express so that we can determine if the prevalence of viral infection targets was increased within the adjuvant cocktail group. Before I finish, I'd like to acknowledge Dr. Adebimpe Obadan for mentoring me in this project, Professor Deb Fuller for facilitating all this research, Dr. Kenneth Bagley for designing the DNA vaccines used, Thomas Lewis, Jim Fuller, and Dr. Sandra Dross for their contributions to this research, all of the animal research staff at the Washington National Primate Research Center, and the National Institute of Health for funding this research.